What is up, BioClass and W? Today we'll be talking about the techniques of swabbing, gathering bacteria samples, and most importantly, looking at your bacteria samples. Now off to my partner.
Have you ever wondered what's in the bathroom door handle and what kind of bacteria is living there? What about the corner of the cafeteria where there's a mold been growing for the past decade? Today we're gonna find out.
Victor, get the hell out!
Scientists use a variety of different mediums to grow bacteria. Think of each medium as a different tool in a toolbox.
The first type of agar you can use is nutrient agar. It is great for growing a wide variety of bacteria. It's kind of like a all-you-can-eat buffet. Off to my partner.
Another popular type of agar is called matinol salt agar. This one's salty. It's used to isolate Staphylococcus bacteria, like a staph infection, and it turns yellow. And if it turns yellow, it means that the Staphylococcus is present.
And so in methylene blue agar, also known as EMB, it helps detect E. coli, which turns green.
What kind of green?
Metallic. Remember? Metallica! Alright, next thing.
Our next type of agar is called McConkey agar. This agar is used for gram-negative bacteria, and lactose fermenters turn pink. What kind of pink?
Bubblegum pink.
Yeah.
Alright, now, second to last, we have blood agar. This shows how bacteria break down the red blood cells. This can help doctors in the medical field to figure out what type of disease or problem the patient might have.
For example, streptococcus infections are easily identified with blood agar. Lastly, we have Sawbarad agar. This one's for you fungi, like yeast and mold. If a scientist uses this kind of agar, it is easy to identify and detect different kinds of mold, like black mold and mold like in the corner of the cafeteria. Quick question, Matt. How do scientists classify bacteria?
Well, once the bacteria grows, and scientists classify them by using gram staining and their shapes. One of the possible gram stains are positive gram positive, which turns purple. It's like a thick cell wall like Staphylococcus or Streptococcus, and this allows the scientists to know if it's one of those types of bacteria. But there's also other factors that determine.
Gram-negative is another kind of gram staining results. Think of these as like thinner cell walls. While gram-positive presents thick cell walls, gram-negative presents thin. Examples of thin cell walls would be E. coli and Salmonella.
It's pretty interesting, huh? Now, off to other factors like shapes. Saucy, which is round, is found in clusters like Staphylococcus or chain Streptococcus. If you see something that's round and it's gram-positive, you'll know that's one of these main bacteria groups, which will allow scientists to figure out what type of bacteria there is.
Man, I believe it's pronounced cocci.
Oh, that's pretty interesting. I did not know that. Next we have bacilli, which is rod-shaped. Rod-shaped will allow scientists to figure out much precisely what type of bacteria there is, as bacilli has less types of bacteria and it's a smaller and more minimal group, like E. coli is a rod-shaped.
If you've ever seen a model of E. coli like I have, I know Matt, you've also seen a model of E. coli diagram. You can obviously very distinctively tell that E. coli is shaped like a rod.
If you guys want to see one, come by AP Bio class and you'll see one. Lastly, we have spirilla or spiral. This is the Latin word for spiral, if you guys wanted to know. And like Helicobacter pylori, which causes stomach ulcers, you'll be able to know if you have a stomach problem. But also, you'll be able to figure out other bacteria that might be harmful to your body or around you. So, just as a brief summary, you don't want to have stomach ulcers. How many colonies do I need to know to classify a certain type of bacteria and know how much there is?
Well, when you're observing bacteria on a petri plate under the microscope, you have to manually count each colony that is present. It's called direct bacteria counting. If you have 25 to 250 colonies, you have to classify and quantify how many colonies you see. For example, if I'm going through my sample and I count 240 colonies, I would have to put in my data table wherever I'm quantifying the sample number 240. Now, if there's less than 25 colonies, that is not significant bacteria growth, and it is typically considered not significant, and therefore it is not considered bacteria growth. On the other hand, if it's over 250 colonies, all you have to write on your bacteria data sheet would be too many to count. Because even if you can count how many, even if it's 251 colonies, even if you know the exact number, it is technically quantified as too many to count. I was watching a video the other day, Matt, and I saw a concept called antibiotic resistance. What exactly is antibiotic resistance, and how does it occur?
Well, it's when bacteria stop responding to antibiotics. This is called antibiotic resistance, and it's a huge problem for scientists, as it can make the bacteria or disease very hard to treat. One of the possible antibiotic-resistant bacteria are MRSA, or methicillin-resistant Streptococcus aureus. It causes infections, and it resists penicillin, which can cause the body to just be more in pain.
Another example of a drug-resistant bacteria is called VRE, or vancomycin-resistant Enterococcus. This is a big problem this is a major problem because it can cause urinary tract infections and bloodstream infections, which you do not want.
Another one is multi-drug resistance, Pseudomonas aeruginosa. It's found in hospitals, and it causes lung infection. This means that even though you think hospitals might be safe, they really aren't, so look deeper.
Number 4, we have carbapenem resistant
Klebsiella pneumoniae, or known as CRKP.
These are hard to treat because these are really a major problem. This bacteria is a major problem because it can make it hard to treat pneumonia and other urinary tract infections. Pneumonia is a major and very common lung and respiratory issue, and this antibiotic-resistant bacteria makes it even worse.
Oh, that's good to know. Well, 5th, we have XDR-TB, extensively drug-resistant Mycobacterium tuberculosis, and it makes tuberculosis nearly untreatable, which means you're on the verge to die, buddy.
You know what, Matt?
Good luck.
I bet Arthur Morgan had this drug-resistant bacteria because Arthur Morgan unfortunately contracted tuberculosis.
Some of these are not like techie people.
Number 6, we have Neisseria gonorrhea,
or known as super gonorrhea.
How many booms does that get, Matt?
Boom, boom, boom. 3 big booms. And it's becoming resistant to last resort antibiotics, which means that's going to soon become untreatable, which means it can show how genetically it is becoming stronger and it is becoming more resistant, which shows how it is evolving, and that means that we need to evolve as humans and make stronger antibiotics.
Exactly. I mean, last resort antibiotics are the last line of defense we have against these kind of bacteria and against these infections. These bacteria come in, they're already resistant to it, and this is just, even though this is the last resort antibiotics, they're your last chance to be healed, and these bacteria make it so that's impossible.
Yeah, just as a final thought, these bacteria can mutate, share resistance genes, which is very bad, and it may even make more bacteria resistant to these antibiotics, which shows that we should try to get rid of these bacteria so that there are less antibiotic-resistant bacteria. And they thrive when antibiotics are overused, which shows that sometimes antibiotics might be bad for your disease. So how do we fight back on? Do you know?
Well, Matt, doctors are fighting back with new strategies to combat antibiotic-resistant bacteria. One way that the doctors are fighting back, scientists are fighting back, are better antibiotic stewardship. This is a concept where doctors only use antibiotics when they're truly needed. This helps to make it sure that the bacteria cannot adapt and become resistant to the antibiotics because they're not exposed to it. When we overuse antibiotics, the bacteria in the body in an infection become resistant to it. So if we use less antibiotics, this will lower the chance of bacteria becoming antibiotic resistant.
Oh, I also think I know one. It's like called combination therapies, right? It's using 2 or more drugs to stop resistance, which shows it's basically like from both sides, and the bacteria can't hold it off.
Yeah, if the bacteria is resistant to one of the drugs, maybe the second drug is not resistant.
Or they work as a team and they fight together.
That's good, man. Third type of way doctors are treating bacteria that are resistant to antibiotics is called phage therapy. This is where they actually inject bacteriophage viruses into the patient, and that can help to attack the bacteria directly and physically, that in a way that they cannot be resistant to.
But wouldn't that be
Bacteria cannot be
dangerous?
resistant
Wouldn't
to.
that be dangerous though?
This is very dangerous in immunocompromised individuals, which means that if an immune system is weak and the bacteria cannot be fought off itself, and the viruses can actually attack the body itself, and that would be a big issue. That's why phage therapy is not used very extensively.
Oh, I think they could also just make new antibiotics, but the problem is that takes time and money.
Money is money. To conclude, basically, to slow resistance, doctors and us as humans, we can all help by using our antibiotics and not using them effectively and the way they are prescribed, and not using them for a wide variety of infections. If we have a common cold or a small infection, there's no need to use antibiotics.
Just go to sleep and you'll be completely fine, and drink some hot tea. You'll be good.
All right, so that is the end of our video. Make sure you hit that like button and subscribe to our channel.
And comment what you learned and was new. Bacteria are amazing, but they also can be dangerous. By understanding how they grow, spread, and resist antibiotics, we can stay one step ahead, which is always better for us.
If you like this video, again, hit that like button and make sure to subscribe.
For more science content from Biology Boys. Guest appearance, come in. Cut outro.